Thank you for introduction. I'd like to tell you something about neosex chromosomes and their possible role in radiations of lower ditrysian moths. I should first explain what are lower ditrysian moths. So this is higher level phylogeny of Lepidoptera, and you can see that like 98% of all Lepidopteran species belong to the group called Ditrysia, and then around half or more than half, and probably all species of moth and butterflies belong to this group, Macrolepidoptera. And these primitive lineages, or basal lineages, are then called lower Ditrysia or Apoditrysia, and I will talk about two superfamilies, the most diverse superfamilies in these lower Ditrysia, the Trichoidea superfamily and the Elechioidea superfamily. First, I should give you some brief introduction into Lepidopteran cytogenetics. So here you can see quite typical Lepidopteran karyotype. As you can see, the chromosomes are relatively small, uniform in both shape and size. You cannot identify individual chromosomes. And most frequently, the haploid chromosome number is 31. And it's also thought that this number is ancestral. Well, this is a karyotype of Perlit carop moth, but it's not important for us now. Yeah. And the ancestor karyotype n = 31 is also supported by comparative studies when genetic linkage map of diamondback moth, which is here, which is important pest, or peppered moth, Biston betularia, which is model for industrial melanisms, were compared to the reference genome of Bombyx mori, here, silk moth. So obviously, they differ from Bombyx by the same rearrangements. So supposedly, indeed, at least the common ancestor of this group had chromosome number, 31. And what about sex chromosomes? Well, Lepidoptera, moth and butterflies are the largest group of animals, or largest group with female heterogamety, which means that female has female-specific W chromosomes and male has 2 Z chromosomes. What are another important and quite unique features? That females have achiasmatic meiosis. That means that there is no recombination during female meiosis, and female-specific W chromosomes is full of repetitive sequences, and probably there are no genes, or at least there are no known sequences, coding sequences from W chromosome. And also there is no dosage compensation between male and females. It will be important later. This is another karyotype of codling moth, Cydia pomonella from the family Tortricidae. It's our main model in our lab, and you can see that the chromosome number is reduced to 28, haploid number 28, and there are 3 pairs of extra large chromosomes, and conspicuously the largest one was identified as a sex chromosome pair. And it's interesting because such large chromosome pair was observed also, or reported also in other species of tortricids, and it was also predicted based on some anecdotal linkage data that there was probably translocation between sex chromosomes and autosomes in common ancestor of Tortricidae. So in order to test this prediction, we decided to perform physical mapping of sex chromosomes, or of Z chromosome of codling moth, using so-called backfish. What is backfish? It's fluorescent in situ hybridization with bacterial artificial chromosomes as a probe. Bacterial artificial chromosome is just plasmid with large piece of DNA, like 100 kb of genomic DNA. You select the the clone containing your gene of interest. You then label it by fluorochrome, denature it, and hybridize it to the nature chromosomes, and then it will label the physical position of your gene on chromosome. And because you are limited by number of fluorochromes, or more likely by your fluorescent microscope and number of filters, you can repeat the hybridization with different sets of probe 3 times max. And then when you merge your results, you have physical map of your chromosomes or part of it. These are orthologs of Z-linked genes or genes Z-linked in Bombyx mori, and you can see that they hybridized 1/2 of this chromosome. I should say these are not mitotic chromosomes; these are meiotic chromosomes. It's packed in bivalence. That means that homologous chromosomes are paired together. And yeah, here we have a whole nucleus. It's again packed in nucleus with different set of probes. This time, these are genes or orthologs of genes from chromosome 15 of Bombyx mori. And again, you can see that they hybridized 1/2 of the bivalent. Here, there is overlap. We have here hybridized chromosome, ribosomal protein P0 and shaker, and they both hybridize to single chromosomes. And this is female nuclei or nucleus, and you can see this is WZ bivalent, and the signals are only on one thread of the bivalent, which means that they are maternally inherited copies or alleles, probably degenerated due to restriction of recombination. And here, when you measure relative position of signals, you can make such a nice scheme. So you can see again that really Z chromosome of Ahdiahla pulmonella consists of chromosome 15 and Z chromosome of Bombyx mori. And we were interested in the origin of this rearrangement. So we used quantitative PCR to, in fact, determine the copy number of Z-linked gene in relation to autosomal reference. If the gene is autosomal, then the ratio between in males and females should be 1:1. If it's Z-linked, then male should have twice as much copies as a female. And here it is, we developed this technique or tried this approach in codling moth, and then we use it in 2 other species of tortoisid. One from is Lobesia botriana, from the subfamily Oleutretinae, the same as Ahdiahla pulmonella. The other one is from subfamily Tortricinae. And as you can see on these 2 genes, acetylcholinesterase 1 and Notch from chromosome 15, these 2 genes are sex-linked in all species, which means that chromosome arrangement probably occurred in the common ancestor of these 2 subfamilies, and these 2 subfamilies comprise like 97% of all tortoisid species. And also, if you look at the chromosome number of these species, so in Tortricinae, like modal chromosome number is 30, haploid chromosome number is 30, which means that chromosome, sex chromosome autosome fusion was probably the first arrangement which differentiated the common ancestor of these subfamilies from the ancestor karyotype. So it poses question on or about the possible role of this rearrangement in radiation of these subfamilies. And well, I found very inspiring some papers on ecological speciation and neosex chromosomes. For example, in stickleback fishes, it was shown that turnover which means rearrangements of sex chromosomes, might promote speciation. And in birds, it was suggested that gene content might be relevant in no-sex chromosome maintenance. So if we look into the, or if we look at the gene content of these two chromosomes, Z chromosome is known for its role in polyzygotic reproductive isolation. It harbors disproportionately high number of genes underlying differences between closely related species in Lepidoptera. Also, in some species, female host preference was shown to be Z-linked, and also some components of the chemosensory system are Z-linked in Ostrinia nubilalis, which is like model for chemoreception in Lepidoptera. If we look at chromosome 15, it was shown that in Bombyx mori, chromosome 15 harbors two clusters of carboxylesterases and largest or major cluster of ABC transporters. These are gene families involved in detoxification and regulated absorption of plant secondary metabolites. So these are, in fact, performance genes because they affect surviving and growth of larvae on their host plants. And what could be the concept or the advantage of such fusion between sex chromosome and autosome? It was mentioned in the morning that physical linkage between performance and preference or reproductive isolation genes can generate genetic covariance between traits and thus facilitate ecological speciation or specialization and speciation under divergent selection. And we add one more possible mechanism. We hypothesize that following the fusion, this now maternally inherited former autosomal part-bearing performance genes start to accumulate mutations, and these maternally inherited alleles ultimately degenerate into pseudogenes, and this could cause or will cause environmental stress, and therefore there could be or should be selection for some compensation. And as I said, there is no global dosage compensation in Lepidoptera. But it was shown, for example, in case of metabolic resistance to insecticides, that such stress could be solved by amplification of detoxification genes, these performance genes. And after this amplification, some of these genes could acquire new function, and it could increase detoxification capacity of larvae and enable them, for example, some host shift or broadening of host spectrum. And it was really shown in swallowtail, papillary swallowtails, that amplification and functional diversification of cytochrome P450 monooxygenases can serve as a key innovation, which in detoxification of secondary plant metabolites, which overcome host plant defenses. So it could really enhance somehow the radiation of the group. And similar observation was made also in superfamily Galechioidea. I mean, this is karyotype of Tuta absoluta, tomato leaf miner moth from family Galechidae, and you can see that again there are some there is conspicuously large chromosome pair, and similar chromosome pair was observed in several groups across the superfamily Galechioidea, and we were interested if it could be sex chromosomes, so we did genomic in situ hybridization, and which means that we hybridized labeled female DNA with male unlabeled DNA to the chromosomes. And as you can see, this chromosome was highlighted by female DNA, which means that it's female specific, so it's W chromosome, which suggests that the other one is Z chromosome. So again, in this group, sex chromosomes are the largest one, so it suggests that there is, again, some arrangement between autosomes and sex chromosomes, and possibly it occurred, again, early in their evolution, or in the evolution of the superfamily Gelachioidea. So it might be some more general phenomenon which we want to study more. And we are interested in the origin and in common features of the autosome involved in these rearrangements. And I'd like to thank the organizer for giving me the opportunity to talk to you, and also to you, the audience, and to my collaborators or colleagues from our lab of molecular cytogenetics, led by Professor František Marec, and to our colleagues from the United States, Lisa G. Nevin, who provide us PAC library, and Ken Sahara from Japan, who taught me backfish, and that's all. Thank you for introduction. I'd like to tell you something about neosex chromosomes and their possible role in radiations of lower ditrysian moths. I should first explain what are lower ditrysian moths. So this is higher level phylogeny of Lepidoptera, and you can see Lepidopteran karyotype. As you can see, the chromosomes are relatively small, uniform in both shape and size. You cannot identify individual chromosomes. And most frequently, the haploid chromosome number is 31. And it's also thought that this number is ancestral. Well, this is a karyotype of Peralid carp moth, but lower ditrysia or apoditrysia. And I will talk about two superfamilies, the most diverse superfamilies in these lower ditrysia, the Trichoidea superfamily and Gelachioidea superfamily. First, I should give you some brief introduction into Lepidopteran cytogenetics. So here you can see quite typically that like 98% of all Lepidopteran species belong to the group called ditrysia, and then around half or more than half, and probably all species of moth and butterflies belong to this group, Macrolepidoptera. And these primitive lineages, or basal lineages, are then called, it's not important for us now, and the ancestor karyotype n equals 31 is also supported by comparative studies when genetic linkage map of diamondback moth, which is here, which is important pest, or peppered moth,